I don't know about you guys, but I got a big old storm over top of me right now. So we'll see what happens. I don't even know where Belton is, Christina. Where is that? All right. Okay. Yeah. Rain equals nice grass, save on the water bill. All right. Any questions about anything so far? We're missing, looks like we're missing 6 students. Any questions? Oh, so everybody knows everything. That's fantastic. Good to hear. So nothing, huh? No questions about nothing? Okay. Gravity is important for blood pressure. What if you're laying down? If you're laying down, your blood pressure is going to be much lower. And I think I explained this last week. Remember, your heart has to pump blood to your brain. And so it's fighting gravity. And so if you're standing up, I don't want to get into the physics here, but if you're standing up, then your heart has to pump blood straight to your brain against gravity. If you're lying down, gravity is not a factor because now you're on the equal footing. Your heart and your brain are on the same level. Does that make sense? Okay. All right, so any other questions about anything? Yeah, we will, Christina. On my syllabus, I said I need a week, so I'm just about done, and so we will go over the exam on Wednesday. All right? It's not easy grading 24 exams that are all essay, especially trying to figure out exactly what you're saying. So we will talk about it on Wednesday, okay? Anything else? No one has anything about the previous. All right. Okay, then we're going to move on. All right. So, I remember I told you before, I'm not going to make a big deal about capillaries, but we'll talk more about capillaries when we talk about the respiratory system. Because like I said before, it's all connected. All right? So there's just a couple things I need you to worry about for the next exam. And that is exactly what's going on as far as transport. What's going into the capillaries, what's going out of the capillaries. Don't worry, I'm not going to ask you about a atrial venous anastomosis. That's not important, all right? So you can just save yourself. That just means a bypass, all right? That's all that means is a bypass. Not important. What is important is understanding that there's a lot of things moving in and out of a capillary based, again, on its solubility, its charge, its polarity. We do have pores and clefts, so things are going to leak out of the capillaries, all except for what? This is going to be a recurring theme from here on out. What normally would not pass through a membrane? Somebody. Okay, Bucola, very good. Diani, beautiful. Proteins, because they're too big. All right. Beautiful, 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 beautiful. All right. So again, if it's something that's lipid soluble like CO2, O2, or steroids, things like that, they're going to go right through. We have others that are going to be transported in some way, form, or fashion. All right. Now, this is the only tricky slide right here. Again, hydrostatic pressure just means that because of typically of gravity and water pressure, you're going to have things pushed out. All right, so don't let that word freak you out. Hydro refers to water, and static, we're talking about gravity here. So things are constantly leaking out of your capillaries, your plasma, not your blood cells. That would be a bad thing, unless you have some sort of damage. All right, but the plasma is going to leak out, except for the proteins. The proteins aren't going to leak out. So we have a thing called colloid osmotic pressure, and this is important for your next test. So write it down. The plasma leaks out. It contains everything that was in the plasma except for the proteins. So, good old-fashioned osmosis, remember that? Osmosis says that water is going to diffuse across the membrane from an area of low substance concentration, in this case proteins, to an area of high. So the water is going to want to come back in. So this is kind of what's going on every day of your life. Plasma leaks out of your capillaries. The water comes back in due to this colloid osmotic pressure. What's left of that fluid becomes what we call lymph. And this is, I'm going to go through this real quick because we're going to talk about the lymphatic system on Wednesday. So one of the many functions of the lymphatic system is to collect all the stuff that leaked out of your capillaries. It will now enter the lymphatic system and eventually be brought back into the circulatory system. So again, don't worry about this slide as far as the lymphatics and the vessels and all that stuff. That'll get a full discussion on Wednesday. So, and again, don't have to worry about these numbers. This is just when you're like, oh, wow, that's cool. So if we look at your typical average here of cardiac output of 5 liters per minute, 60 minutes, 24 hours a day, 20 liters of plasma leaves the arteries. 17 liters comes back in through osmosis. 3 liters are actually carried away to the lymph, where they'll get a check. That's where we check to see if you have any bacteria, any virus-infected cells, any cancer cells. Again, all stuff for Wednesday. And then it will go back into the circulatory system that we'll talk about later. So these 3 slides, 1, 2, don't worry about it until we get to the immune system. All right, here we go. This is the regulation stuff. And you know there's going to be several essay questions on regulation. So again, intrinsic and auto-regulation mean pretty much the same thing. This is what's happening at the local level, all right? In terms of what we're going to call vasoconstriction, vasodilation. Extrinsic, once again, is going to involve either the nervous system or hormonal control. So your auto-regulation is based on what's going on at the tissue. And I want you to kind of focus in on your skeletal muscle for the most part, all right? So, and I want you to focus on this when we say metabolism increases, we're talking about exercise or hard work, all right? So when you're exercising or doing hard work, your O2 levels are going to decrease because you're using oxygen for what? What are we using the O2 for during exercise? Somebody, just so we're on the same page, I'm looking for 3 letters. No, not muscle contraction. I need O2 to what? Thank you very much, Andrea. ATP, cellular respiration. And of course, you should know that one of the byproducts of cellular respiration is carbon dioxide. So when we're exercising, our O2 levels are lower because I'm using it, and my CO2 levels are higher because I'm making it. If I'm exercising hard, I'm going to produce lactic acid. That's going to decrease my pH. My levels of adenosine is going to increase because I'm using ATP. Now, who can impress the daylights out of me? Why would my potassium levels be higher outside the cell in the interstitial fluid during heavy exercise? Can anybody put that together? Why would my potassium levels be higher? Your hint is action potentials. Crickets? No one? I have lots of potassium outside the cell. Your hint is action potentials. Why? What about the potassium pump? You're leaving a little bit out of, is it just the potassium pump? The what pump? What's our pump, folks? See, this is why I know I'm harping on it. Okay, Christine. How you feeling, Christine? You okay?
I'm dying very slowly.
What's that?
I'm dying very slowly.
Oh, I'm so sorry to hear that. Okay, so Christine's dying. We'll see how long she lasts. Okay, so yeah. Somebody, I'm going to pick a name. Yeah, there's Katie with her evil thoughts. No. The hint was action potentials. So once again, who do I pick? Sarish. What's the role of potassium in action potentials? What's the role of potassium in any action potential? All right, very good. Repolarization. Okay, so 2 of you got that, repolarization. So again, every time I create an action potential, I have to repolarize, and I do that by opening potassium channels and letting potassium out. So if the sodium potassium pumps aren't keeping up, we're going to have extra potassium outside because, again, the sodium potassium just can't get it in fast enough. All right. Like I told you, I warned you, a lot of stuff that you learn early is going to come back over and over again. All right. So this is a good study slide. Make a note of it. So again, we're talking about intrinsic things at the local level. We haven't talked too much about endothelium, but that is, again, a chemical that your endothelium produces, typically when there's damage, to cause vasoconstriction to reduce blood flow and ultimately blood loss. I'd like to think that everybody knows that cold application is going to cause vasoconstriction. Right? We all know that? Do we know that? And exactly how does that work? If you sprain your ankle, you're going to put ice on it, right? Okay. Andrew wants it recorded. All right. I'll record it. So I'm going to make a new rule. It looks like we're still missing 4 students, but so here's the new rule. I'll record these and make them available as long as I have at least 75% of the class attending the lecture. Does everybody understand that? Because like I said, my concern was people say, I don't have to go to class. So as long as I have 18 students or more, I will record the lectures. All right? Deal? Okay, where were we? Oh. Okay. All right, fine. So yeah, we put, if you sprain your ankle, you're going to have inflammation, which we're going to talk about on Wednesday, which is bad. And so if we can reduce blood flow to that area, we can reduce the leaking out of the plasma. And so, yeah. Of course, that's our application aspect. We do that, and we'll talk about this on Wednesday too. But typically, when it's cold outside, you want vasoconstriction to conserve your body core temperature. We don't want blood going to our fingers and toes, because that's just going to radiate out. And then we have to worry about what? What do we have to worry about if it's cold outside and you're not well protected?
Frostbite.
Yeah. But we're trying to avoid hypothermia. So, and this is just a kind of cool little thing. So your body says, hey, you know what? I don't want to die. I can't have hypothermia. I don't want my core temperature to go down. But the only way I can avoid that is to cut off blood flow to my limbs. So you get frostbite, which means you reduce blood flow to your fingers and toes as your body tries to conserve the body core temperature. All right, enough of that. And of course, the vasodilators include nitric oxide. We've talked a little bit about that. But again, that's going to cause vasodilation. Heat, again, does the opposite of cold, vasodilation. Histamine, which we'll talk more about when we get to the inflammatory response in the immune system, and all the things that we just talked about a little bit ago. Samantha asked, could you give an example question of how this slide will be used? Well, it'd be really easy, right? And like, give me 2 intrinsic vasoconstrictors and 3 vasodilators. How simple is that? Yeah, easy breezy. Okay. Let's press on. Okay. And then we have something that's this is intrinsic as well. It's at the local level, and it's called myogenic control. Myo referring to muscle. Genic means originating in the muscle. And again, this is just the characteristic of smooth muscle. And I know I mentioned this a little bit earlier. If you stretch smooth muscle, it will automatically contract. So if my blood pressure gets high, it's going to dilate the vessel, and the smooth muscle in the vessel is going to turn around and contract. And vice versa. All right, if the pressure is low and it's not stretched, then it's going to relax and dilate. All right, so that's part of the intrinsic thing. See, between this and this, those are your two slides to study to give me examples of vasoconstriction and vasodilation intrinsically. Okay. Extrinsic, like I said, involves the nervous system and the endocrine system. And again, you break this word down so it doesn't sound so foreign. Sympatho refers to the sympathetic nervous system, and adrenal refers to the adrenal medulla. And again, remember, under sympathetic response, your adrenal medulla is going to release epinephrine and norepinephrine, and that's going to divert the blood like we did way back when. Where do we want the blood to go? We don't want it to go to the skin or the digestive system or the kidneys. We want it to go where? Skeletal muscles. Very good. All right. Very good. And that was that slide way back when. I'm going to pop it up right there. So boom. Yeah. So here you're relaxed. All right. 15-20% blood going to skeletal muscle. Heavy exercise, 80-85% to the skeletal muscle. And that's what that sympathoadrenal refers to. We're just going to change where the blood is going. All right, let me get back to where I was. Boom. All right. There we go. Parasympathetic is really not involved. All right. If you don't have a sympathetic response, then the local things are going to take care of your blood flow for the most part. All right. The only really active role, and we will talk more about this, is the parasympathetic comes in to play for vasodilation, obviously in the digestive tract for rest and digest, and for the external genitalia, i.e. our erectile tissue, which we'll talk more about later. All right. So this is just kind of a summary slide when we talk about blood pressure regulation. Remember, MAP refers to that mean arterial pressure. Anybody remember that number? What was that number on average? If you're relaxed, what's your average arterial pressure? Oh, no. That's your cardiac output, Diani. What's your pressure? It's that little formula. Well, that's your normal blood pressure, but what's the average? Remember, that was a little formula that I covered. All right, Daniel gets the gold star. All right, 93. Remember, that's going to be on your test. Remember, MAP is equal to your pulse pressure, which is systolic minus diastolic, plus a third of your pulse pressure added to your diastolic. Go back and look at that. All right. Okay. Beautiful. So your MAP, your pressure is equal to your cardiac output, and TPR, which refers to total peripheral resistance. This means vasoconstriction. And so we know that your cardiac output is a function of heart rate and stroke volume. So these three things are the most important factors in dictating your blood pressure. If we increase heart rate or stroke volume or both, and we have more vasoconstriction, then we're going to increase our blood pressure. All right, moving on. So again, for short term, remember this is your nervous system. Your first responders, you have baroreceptors and chemoreceptors. They're found in your carotid artery and your aortic arch. There's little centers there that are monitoring your blood pressure and the composition of the blood in terms of O2, CO2, and pH. All right. So, oh God, I love this slide. What is so cool about this slide? Somebody. Come on. Talani, that's not what I wanted to hear. It's an exam. What's so cool about this slide? Anyone? It's definitely on the exam. Okay, Bukova, that's not bad. Homeostasis. What I love about this slide is it encompasses a lot of the stuff that we've covered. It is indeed a negative feedback, but what I wanted you to get out of this is a slide that puts a lot of stuff that we've covered in this course all together. And so here's where I'm going to quiz you. I'm going to go by name. All right. So let's just take one. Let's say we have, excuse me, high blood pressure. So I have high blood pressure and I have my carotid body baroreceptors as well as in the aortic arch baroreceptors. They're going to detect that high blood pressure and they're going to send that information back through our, and I love this so much, our afferent pathways, which includes the vagus nerve, which you all are supposed to know, and then we have also the glossopharyngeal nerve. And it's going to go to the regulatory centers in the medulla saying, hey, blood pressure is high. So the medulla will send info via our efferent pathway, the vagus nerve. Remember, this is a 2-neuron pathway because we're talking about the autonomic nervous system. So let's see if anybody remembers their stuff. Who do I pick? Let's see here. Joshua. What neurotransmitter is my parasympathetic preganglionic neuron going to release on the postganglionic neuron? What neurotransmitter from the preganglionic parasympathetic neuron going to release on the postganglionic? We'll give Joshua 10 seconds, and then anybody can chime in to help him.
Would it be acetylcholine?
It would indeed, Joshua. Excellent, 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 excellent. Acetylcholine. All right. The postganglionic parasympathetic neuron is going to release what neurotransmitter? Anybody? Just throw it out there. Okay, good job, Jennifer. Good job. Yes, excellent. Acetylcholine as well. Now, for a gold star, what receptor is going to bind with that acetylcholine on the heart? Who's going to be a hero?
The muscarinic receptors.
Okay, Nada, very good, but can we be more specific? Anyone? Come on, you can do it. No, no. We're releasing acetylcholine, and it's on a muscarinic receptor. We've established that, but which muscarinic receptor? This is all testable stuff. No, Daniel. Betas are adrenergic. They're going to bind to epinephrine. Yes, Jennifer.
Is it muscarinic 2 receptor, which is located?
Very good. Very good. M2 receptor. Very good. All right. See, this is a perfect example of how you can't forget the stuff that we've already covered. So yeah, acetylcholine will bind to the M2 receptor. When acetylcholine binds to an M2 receptor, it's going to slow down the heart. How is it going to do that? Who do I pick on now? Who do we pick? Somebody that I haven't heard from in a little bit. Gabriella. When acetylcholine binds to an M2 receptor and I want to inhibit the heart, I'll give you a hint. What channel opens on the cardiac muscle cell? Anybody? Okay. Good job, Cindy. Good job, Daniel. Good job. Excellent. Yeah, we open potassium channels and potassium leaves. Okay. All right. And real quick, if my blood pressure is too low, once again, the baroreceptors will send a signal to the glossopharyngeal and vagus nerve, and now my excitatory centers will fire up and send signals through our sympathetic nerves. And one more time, let's see if we can get this, because this is a big test question. So the preganglionic sympathetic neuron is going to release acetylcholine, and the postganglionic neuron is going to release what? What's it going to release? All right, norepi, any works, beautiful. And that's going to bind to what receptor? Very good, Daniel. Beta 1. And that's going to open sodium and calcium channels. So again, the whole purpose of this whole slide will show you how we put this all together. All together. All right, let's move on. This slide shows you the same exact same thing. It was kind of redundant, but again, instead of pressure, we're looking at O2, CO2 levels, and pH, which we'll talk about later. And this is just a big, wordy slide that covers everything I just said. Okay, now we're looking at our endocrine system. You see these two bolded. What do you think that means? Oh, look what I just did. All right. Okay. Beautiful. So you're gonna be tested twice on this. Once in this lecture on our vascular system, and next time you're going to see the exact same information on the kidney, renal stuff. So here we go. You have to be able to explain this. You use short sentences. If we were in class, you would draw this, which saves you a little bit of time. But here we go. This is called the renin-angiotensin-aldosterone system. So your amazing kidneys, which we'll talk about quite a bit next week, have unique little cells that detect when your blood volume and your blood pressure are low. So right now you just have to remember JGA, juxtaglomerular apparatus. You don't have to write out juxtaglomerular apparatus, just remember JGA. Those cells are going to detect when your volume, your blood volume and blood pressure are low, and they're going to release renin. Renin is an enzyme that's going to convert angiotensinogen, and this stuff is circulating in your blood right now as we speak. That was made by the liver, and it's circulating. So when your blood pressure drops, the volume drops, renin is released, and it's going to convert angiotensinogen into something called angiotensin 1. The next step is there's an enzyme produced by the lungs. And again, this is why I want you to see how everybody's working together here. So you probably would have no idea how the lungs and the kidneys actually work together. But they do. So the lungs produce what's called ACE. ACE stands for angiotensin converting enzyme. Angiotensin converting enzyme. ACE converts angiotensin 1 into angiotensin 2. Angiotensin 2 is a very powerful hormone. It does two important things. It causes vasoconstriction. That's that TPR, total peripheral resistance. I want to cause vasoconstriction to help increase my blood pressure. And here's a more complicated part. Angiotensin 2 gets the adrenal cortex to release aldosterone, which we've already covered in the endocrine system. Aldosterone gets the kidneys to retain sodium, and water follows. And you're going to hear me say this numerous times, so never forget. Wherever sodium goes, water follows. Osmosis. Osmosis. Yeah, so Katie just mentioned ACE inhibitors. I'm sure some of you have heard that term. And so think about it. What is an ACE inhibitor? An ACE inhibitor is a drug that knocks out the angiotensin converting enzyme. If I knock this out, I can't convert angiotensin 1 into angiotensin 2. So this is an attempt to keep your blood pressure low. Everybody got that? Does that make sense to everybody? So that should be another aha moment for you. An ACE inhibitor ensures that angiotensin 2 is not created, so I don't have these mechanisms to elevate my blood pressure. Okay, the next important one is the vasopressin, otherwise known as ADH, antidiuretic hormone mechanism, and this involves your hypothalamus. Question. I don't know what that means, Cindy. The slash there, but that's okay. So your hypothalamus also is going to detect or get information about your blood pressure, from those baroreceptors and the hypothalamus. And again, this is where it all comes into play. The hypothalamus will get the pituitary, specifically the posterior pituitary, specifically the neurohypophysis, to release vasopressin, which we've already covered. Vasopressin, we learned that it is an antidiuretic hormone, getting the kidneys to directly reabsorb water. And we'll see more about that when we talk about the kidneys. So I'm reabsorbing water. I'm not going to let it go or be excreted. And it also causes vasoconstriction, increasing that to total peripheral resistance. Okay? So you're going to see the same slide, both of these, or all three of these, on the renal system as well. All right? You need to know this. This is, like I said, high blood pressure is a big problem in this country. So you need to know exactly all the mechanisms involved here. All right? And again, this hormone has two names because it has two different functions. Antidiuretic hormone is involved in retaining water. Vaso refers to blood vessel, pressin to constriction, so vasoconstriction. All right. Now, don't let this next slide scare you. You don't need to know any of this stuff. Atrial natriuretic peptide is a hormone produced by, believe it or not, the atria when the pressure is too high. When the atria are stretched because I have too much venous return, if you will, it's going to release this ANP, which cancels out, and that's all you need to know, is this. It cancels out the renin, the ADH, the aldosterone. That's all you have to tell me. Okay? You don't need to know any of this other stuff. Don't freak out about all this stuff. All you're going to tell me is ANP is antagonistic or stops all the mechanisms involved in increasing blood pressure. And last but not least, your brain, remember, it has the blood-brain barrier, so it's not going to be influenced by neurotransmitters or hormones. It's going to work autonomously, if you will, locally. So it's going to use that myogenic thing, where if I stretch the smooth muscle in a vessel, it's going to turn around and contract. And the metabolic thing that we talked about with the increased potassium, increased CO2, increased acid or hydrogen ions, increased adenosine. So that's at the local. And I got to do some research. So yeah, so that's how we have a lot more sophisticated things. But way back in the day, we used to try to determine what parts of the brain were responsible for different functions and roles. So we could take a radiograph here while you were doing a particular task, and we could see where most of the blood was going. Very cool stuff. Very cool stuff. So if I asked you to draw something, and you're drawing something, and we look at your brain, we can say where most of the blood is going. Pretty cool. Any questions? Any questions? No questions? Okay. That wasn't too bad, was it? Of course, like I said. Sorry about the dogs. Benny, shut up. That's my pug. All right, I tell you what, let's take a 15-minute break, and we'll come out and knock out the blood. So come back at 6:35. All right, 6:35. Same place, yeah. Wait, no, no, the next one will be titled Blood, since we're recording that. So it'll be a new room that will be for blood. Okay? Okay, welcome back. Yeah, I don't have any rain either. I got a lot of thundering, lots of thundering. So, while we wait, we're missing about 4 people. Actually, more than that, like 6. But here's your Gee Whiz fun fact. If you look at Katie's little chat, she says a cat and dog falling from the skies. How many of you've heard the term, it's raining cats and dogs? Does anybody know where that phrase came from? No idea, huh? No? Good guess, Delati. I'm into these kind of things because we have so many phrases and they must have came from somewhere. So here's your little story. Way back, and I'm talking way back, in the olden days, when people lived in thatch hutches, just a basic, very basic hut with a bunch of straw on top of the roof. Those that had pets, the cats and the dogs, would like to sleep on top of the straw that was on top of the roof. So when it rained, the cats and dogs would jump off the roof to get out of the rain. And that's where that term came from. Isn't that cool? Yeah, that's a true story. So it was raining hard and you opened your door, you'd see all these cats and dogs jumping off the roof. And that's where it came from. Okay, we're still missing a bunch of people, but we will move on. All right, so the good news is, when we talk about blood, there's very little about gates and channels and this and that, but there's a lot of numbers that you need to know. So here we go. So like I said, this is pretty straightforward. This is mostly memorization, folks. So not a little bit of physiology, but not a whole lot. All right. So, yeah, I'll record. Like I said, there's the new rule, and since there's 24 in the class, I need at least 18. So keep that in mind. If you decided to blow this class off, you can't expect that they'll be recording, because if I go below 18, it will not be recorded. All right, here we go. Why I dropped that? All right, here we go. Like I said, a lot of numbers.
Hi,
So
Andrea.
we're just wrapping up the
Paula,
entire circulatory system.
Serge.
We know all about the heart.
Try to
We know
join
all
the
the blood
new
vessels.
room.
So the last
You have to
part is
exit out of
the
this
blood. And we kind of know all this stuff already as far as the functions in terms of oxygen and CO2, nutrients, delivering hormones. We know all that stuff. We've already talked about it. This is just a refresher slide to know that this is our pathway. O2, nutrients like glucose and amino acids will go from the plasma across the interstitial fluid into the cells, and the cells will produce CO2 and metabolic wastes like urea and uric acid, and that will be sent to the bloodstream to be removed. All right. So again, a lot of the stuff will be discussed when we talk about the kidneys, especially this pH and whatnot. The body temperature, I've already talked about. If it's cold outside, your blood vessels constrict to maintain core temperature. When it's hot outside, the blood vessels dilate to try to get rid of all that heat. The disease we'll talk about on Wednesday, and we will talk about blood loss today. All right, so some of this stuff is going to be the exact same thing that you will see in the immune lecture. All right, so you'll get a double dose of that. So when we talk about the blood, we have the plasma, which is the liquid portion. It's mostly water. And then our things that we know. Remember, electrolytes is another way of saying ions. So look at calcium, the sodium, the potassium, those types of things. And then we have what is called the formed elements, and that includes our red blood cells, white blood cells, and the platelets. There's nothing new on this slide, so I shouldn't even have to say anything about it. So one of the things, unfortunately, that we can't do because we're online is do what we call, checking what is called your hematocrit. Down here, this is an important term, folks, hematocrit. That is how much of your blood is plasma versus how much of it is the formed elements, primarily the red blood cells, as you're going to see. So this little test tube here was created when, and this is what, again, unfortunately, you would have done in the lab, hands-on. We would have taken a sample of your blood, put it in a tube, and then put it in a centrifuge, spin it around, and separate the plasma from the red blood cells. And then we can determine what percentage, if it is just red blood cells versus what it's plasma. And we'll look at those numbers later. Remember, there's going to be a lot of numbers on this. All these numbers are important. So again, we've already said that your blood volume on average is 5 liters. And the red blood cell count is 5 million red blood cells per microliter of blood. Your white blood cells are much lower, so about 7,500 white blood cells, 5 to 10,000 is the range per microliter of blood. Platelet, 150 to 400,000 per microliter. These are all numbers you need to know for your next test. All right. The plasma, like I said, has some electrolytes and then some proteins. The dominant proteins are your albumins, which I'm sure I've already told you. Those are usually your transporters. They kind of help, excuse me, your lipid molecules, because we don't want them to coalesce. Globulins is another way of saying antibodies. And fibrinogen, which we're going to learn about more today. Now, I don't want you to get in the weeds here. So basically, all you have to say is the plasma is 90% water, 7% protein. And everything else is what we talked about: glucose, ions, hormones. We're going to talk more about these All right. So, oops, sorry. So again, this is just re-emphasizing that we could do this in lab to determine exactly how much of your blood is plasma. And as I already mentioned, those are your primary proteins in there. You have your formed elements, the red blood cells, white blood cells, and the platelets. Again, those are the numbers you need to know for the test. So study those numbers. All right, a little bit about red blood cells. They are very unique shape, what we call a biconcave disc. It gives them flexibility, and hopefully you guys know this, but anytime I have curves on a membrane, what is that for? It's on the slide, but I want to make sure everybody's here. Anyone? Surface area. Very good, Christine. Good answer, Christine. Not bad for somebody that's on their deathbed. So that's good. Gold star, yes. Yeah. Meritorious service. All right. This is just a cool thing. I'm not going to ask you on the test, but think about that, folks. Think about that. Andy's back. Where you been, Andy?
My internet sucks today.
Yeah, probably the weather.
Yeah.
Okay. So think about that. Your body, your bone marrow, to be exact, is producing 2 to 3 million red blood cells per second. What does that also mean, Andy?
That's a lot.
Okay, but what does that mean if you're producing 2 to 3 million per second? Anyone? Come on. If you're producing that many per second, that means what? Very good. Very good. Very good. So yeah, huge turnover in your body. So just a cool number. Okay. And again, some of the stuff you'll see when we get to the respiratory system. So the oxygen carrying molecule is called hemoglobin. We have we have 4 polypeptides, and each polypeptide that makes up this protein contains one heme group. Each heme group can carry a molecule of oxygen, meaning each hemoglobin can produce or carry or transport 4 oxygens. We're going to see a lot more of that when we get to the respiratory system. Your leukocytes, or white blood cells, are much larger than the red blood cells. And again, we'll go into great detail when we talk about the immune system on Wednesday. All right, so we have our neutrophils, eosinophils, basophils, monocytes, lymphocytes. And they get their names on their staining properties and what we can see under the microscope. Daniel, that's a great question. Daniel, you know what? I'm glad you put that on there. All right, Nicola. Daniel, it's the same thing I used to say way back when, was the fact that we all know how to drive a car, but we have no idea what's going on under the hood. Unless you were a mechanic like my dad was. Exactly. All right, so little philosophical stuff going on there. All right, so I'm going to go through these quickly because we'll cover them again on Wednesday. So, oops, I skipped one. All right, so the neutrophils make up, and you do need to know these percentages, folks, make up 60 to 70% of the white blood cells that are circulating through your body. All right, they have a descriptor called granulocyte, which means you can see little granules in there. We don't really care about that. Their job is to, again, defense. So they can phagocytize, they can eat bacteria, destroy bacteria. You're going to have on your lab practical, and this is where histology does come into play. Everybody hear me? Everybody hear me? So you need to be able to identify on your lab practical these white blood cells. So a neutrophil is what we call multi-lobed. It's got all this is the nucleus. All this purple, all the dark purple is the nucleus. So it's got several different lobes. It's called neutral because when it stains, it's kind of neutral. It's kind of in between an acid and a base. Yes, isn't it, Christine? It's pretty. It's a pretty cell. And this is your number one first army. These are the guys that are going to come in there whenever you get an infection, whenever you get any kind of damage, they're going to be there first. Your lymphocytes, what makes them very interesting, and we'll explain again more of this on Wednesday, is all this purple is the nucleus. It's got a ginormous nucleus, very little cytoplasm. They make up 20 to 25% of your white blood cells. And again, they're involved in the immune system, and we'll explain more about those later on. Monocytes, the key characteristic is the nucleus looks like a kidney, if you will. They're very large. They only make up 3 to 8% unless you have an infection, which again will make more sense later. Eosinophils, again, eosine refers to the dye eosin, which is red. And so you can see the cytoplasm stains more red. And again, they're involved in the immune response that we'll cover on Wednesday. Basophils, baso refers to basic blue. And you can see it's almost hard to distinguish between the nucleus and the cytoplasm because everything's blue. And its job is to secrete histamines, which is part of your inflammatory reaction. Thrombocytes, otherwise known as platelets, are actually little pieces of a giant cell called a mega large karyocyte. So you got these scary looking cells that are just shedding off parts of their pieces, and they circulate. They're involved in blood clotting. Since they're just pieces of a cell, they don't last very long. So you're constantly making these little platelets as well. Okay. Pretty easy so far, right? Except for a lot of numbers. So let's talk about, okay, I got it. Andy, what's up, Andy? Andy, did you have a question, or was that just an accident? All right. We'll move on. All right. So, hematopoiesis, or hemopoiesis, if you want to get rid of the syllabus, okay, is again trying to stop loss. Back up a little bit. Here is how we produce our cells. Okay, yeah, I can see how everybody's trying to get back on and off. I get that information where I can see who's trying to get on and who fell off and whatnot. That I can see all that. here we have our pluripotent hematopoietic stem cell or just blood stem cell, and it can differentiate in your bone marrow to make all the different white blood cells and red blood cells. Now, don't study this slide. I'm not going to ask you the flow of how this is different. That's not important. The only one you need to know is the red blood cell. I'll emphasize that in just a little bit. All right. So I'll just give you a couple different versions, but see how it has different names. And this one says instead of pluripotent is a multipotential. It means the same thing, but we make all our cells, our thrombocytes, basophils, neutrophils, eosinophils, they're all made from the original cell. And this is important. It's important. What's the practical application of this? Who knows what leukemia is? Anybody know what leukemia actually is? Andy? Okay. It's cancer, but cancer of what? Actually, Nita, it's the exact opposite of that. No, leukocyte leukemia. So leukemia is a cancer. Remember, cancer in the simplest of terms means uncontrolled cell production. Yeah, Josh was crazy high word, but white blood cell. Yeah, cancer is just uncontrolled growth of cells. The mitosis. Right? And the reason I'm just bringing this up, so there's leukemia, which is you're producing way too many white blood cells. Yeah, it's a cancer in the bloodstream, but we have to be more specific, Kalani. It is the cancer of your white blood cells, the ones that are responsible for your immune response. Okay, so that's where leukemia comes from, because another name for white blood cells is leukocyte. All right, so the reason I'm just bringing this up is if you have leukemia, our best way to mitigate or cure that is to destroy all the bone marrow in the patient through chemotherapy and radiation. We're going to wipe out all their bone marrow and take somebody that has donated their marrow and put it back into this individual. So they get a do-over, because all we need is those stem cells. So when you hear of a bone marrow transplant, we're taking that bone marrow that has those multipotential or pluripotent hematopoietic stem cells and putting it back in there, and then they get a do-over. That's your practical thing. There's nothing on there that was technically testable, but you need to know that. Okay, we're going to move on. I've already mentioned this hematocrit business. This is the ratio of red blood cells to plasma. And these are numbers you have to know for your test. Boom. So for the adult female, 38 to 46%. Adult male, 40 to 50%. And again, the numbers for males on average are higher because of just muscle mass. The more muscle mass you need, the more oxygen and glucose you need to work that muscle, so these numbers will be higher. So for hemoglobin, which carries our oxygen, you need to know these numbers. All right, so there's your range for females and adults, and I expect you to know that. All right, and again, we could have done this in lab. So if you don't have enough red blood cells, if your red blood cells for females, if it's below 38%, you have anemia. For adult male, if it's below 40%, you have anemia. Okay. Polycythemia is too many red blood cells. That is a disease where you're producing more than you need. or that can be artificially done by taking, well, there's a couple different ways to do that. But remember, before I go on to my little story here, remember this is a ratio. So look over here on the far right. If you dehydrate yourself, that dehydration is, again, the water in your plasma. So you can have an artificially high hematocrit because you have low plasma because of the loss of the water. Right? This is bad. Can anybody impress the heck out of me? If I'm dehydrated and I have this hematocrit, what did I affect? What did I affect in terms of resistance to flow? Somebody please impress me. What did I do? We just covered it last week. What are the three factors that involve resistance? Two are relatively constant. Okay, good, Natalia. Blood thickness. What's the term we use? Well, we're not changing the length of the blood vessels. What's the other name for that, Natalia? It starts with a V. Thank you, Joshua. Viscosity, yeah. So that's a bad thing. That's a bad thing. All right. And so take a deep breath, relax. You can put your pen and pencil or paper down because now I'm getting into stuff that I like. So if you look at your hematocrit, and I'll focus on the males, 40 to 50%. In Olympics and in professional bicycle racing and marathons, if your hematocrit is above 50%, you are disqualified because they think you're cheating. Does anybody know what kind of cheating is going on here? Blood doping, not steroids, blood doping. Very good. Very good. And so in the olden days, this might sound kind of barbaric. In the olden days, what athletes would do to cheat is they would give up a pint of their blood. It would be stored. And then let their body, over a couple, 2 or 3 weeks, replace that blood, just like you would if you donated. And then they would actually put the blood they donated back into their body. Because again, the big picture is the more red blood cells you have, the more oxygen you can carry, the more oxygen you have, the more ATP you can have, and now you have an advantage. Christine jumped right into Lance Armstrong. Okay, now Lance Armstrong, that's what they did way back in the olden days, all right? That's not what Lance Armstrong did. Because now we have drugs that you're going to learn about in just a little bit. We have drugs that will stimulate your bone marrow to produce more red blood cells. It's called EPO. How many have heard of EPO? It stands for erythropoietin. And so erythropoietin, it is a hormone, but we also have a synthetic version that we would give to patients that have anemia. So there was a benefit to this drug that we created, but it also would let a normal person that didn't have anemia, if they took EPO, they would produce more red blood cells than normal. Everybody follow me so far? So Lance took a EPO, which cranked up his hematocrit, but he never got caught. He won 7 Tour de France's and never actually got caught cheating by taking EPO. And why? And this is where I'm coming down to this, is remember, hematocrit is a ratio. So let's see how devious you guys are. So let's say you take an EPO and the race time is coming up. They're probably going to take a blood test. What can you do to get your hematocrit below, let's say it's at 55%. What could you do to get it below 50% on a bus before the race? Okay. That would be the opposite, Joshua. If you sweat, your hematocrit would be higher. You don't want to lose water, you want to gain water. Okay, Natalia's onto something. Drink more water. All right, is that going to get you where you need to be before race time? What's a faster way to increase the water in your blood vessels? There you go, Joshua. There you go, Natalie. Yeah. So that's exactly what Lance Armstrong was doing. He was taking EPO, and then right before race time, he stuck an IV in there, cranked up his water level in the blood vessels, and then his hematocrit would have looked fine. Now, I still admire Lance Armstrong, and this is just Jewish stuff that I have to say. I admire, that's good team match, very good, Joshua. Lance Armstrong, I still admire because everybody in that era cheated. So I look at Lance as the best of the cheaters. Right? Yes, I'm a big Tour de France fan, and Tour of Italy, and Tour of Spain. There's the big three. Yeah. So this is a true thing. So Lance was stripped of his 7 Tour de France's, and so there's a big gaping hole in the history of the Tour de France because they normally, if the winner cheats, who do you give it to? Yeah. And if the second place person was also found to be cheated, who do you give it to? Okay. Yeah. So my point is everybody in that era was caught cheating. So there's no winners of the Tour de France the years that Lance won it because everybody cheated. So anyway, that's just my gee whiz little thing. Let's move on. All right. Deani, they didn't know he was doing that until well after he retired, and there was just all these people investigating because no one had ever won 7 Tour de France's. No one had ever done anything like that. So they were just doing a lot of investigating and interviewing people, and finally some people just, they cracked and explained what was going on. All right, I could go on about that. Let's move on. So this is just your gee whiz facts. Yeah, exactly, Joshua. It's all about the money. All right, so this is mostly for you to go ooh and ah. Because if you think of the numbers, Gabriella, can that be exam? Maybe there'll be a bonus question on Lance. Okay, you guys are killing me. All right, so let's move on. Again, a lot of these are just numbers. So, and these aren't, you don't have to know these numbers, all right? I just want you to appreciate what's going on in your body. So one red blood cell has 280 to 300 million hemoglobin molecules. And as I said just a little bit earlier, one hemoglobin can carry 4 oxygens. So one red blood cell can carry 1.12 billion, oh my God, molecules of oxygen. And you think about what I just said, you have 500 million red blood cells per microliter of blood. And so a microliter is a millionth of a liter, and you have 5 liters. And so, anyway, the numbers are just staggering. Basically, your circulatory system is carrying trillions of molecules of oxygen. Again, none of these numbers are testable. I just want you to appreciate that. So we talk about hemoglobin carrying O2, but it can also carry CO2. So we have different names for the hemoglobin. This is not that important at all. All right. So oxyhemoglobin, it's carrying O2. Deoxyhemoglobin, it's not. It gave it up. If it's carrying CO2, it's carb. That's a fun word to say. Carbaminohemoglobin. All right. Again, this is not testable. It's just your gee whiz fact. Carbon monoxide is a problem, especially in winter when people don't understand that you shouldn't have fires in your house or sit in your car and idling. Carbon monoxide is CO. CO2 is fairly harmless. Carbon monoxide is not. Again, this is not testable. It's just for it's your public service announcement. All right. Carbon monoxide loves to bind to hemoglobin. It has 250 times the affinity, which means it can outcompete your oxygen for a site on that hemoglobin, and it stays on there. So people die from carbon monoxide poisoning because, again, the oxygen can't bind, they can't get O2 to the tissues, and you die. All right. That's your GWAS fact. All right. Here's your study slide. Everybody paying attention? And this is the easy version of the differentiation of red blood cell. I expect you to know this for the test. Everybody got it? So we start with our stem cell. We go to a pro-erythroblast. We're making hemoglobin. We get rid of the nucleus. We don't need that anymore. And again, I expect you to know that. I'm not going to read words to you. You can do it. All right. We have a negative feedback. This would obviously be on your test. Anytime we have a negative feedback system. All right. So, very simple. Hypoxia, lack of O2 to the tissues. And your kidneys can detect that. Typically, the natural causes are altitude. The higher you go up in altitude, the less O2 pressure there is. And you could have a disease or something that's going on as well. Your kidneys, again, you probably thought your kidneys were all about just filtering blood and making urine, but they have several functions as you're learning right now. We just talked about the renin-angiotensin-aldosterone system, and now we have the erythropoietin. So there's your kidneys. They notice that their O2 level is low. The kidneys release erythropoietin, and that just stimulates the bone marrow to make more red blood cells to make up for that. Any questions with the feedback? No questions? All right. All right. Okay. We'll move on then. So getting back to our sports thing, I know I have one dog that the Thunder, he just freaks out and he cries like a baby. So anyway, that's Bear. Ironically, bear is the biggest one. He actually looks like a bear. Bear, yeah. Okay. Bear, he's huge. He's like 120 pounds. Yeah, he's huge. He's a very large dog, but he's a crybaby. Okay. All right, where was I? Oh, getting back to cheating in sports. The way you can, how do I put this? The way you can cheat legally is in the Olympics. Anybody know what I'm referring to when I talk about this? There you go. Space Force. Shut up, Katie. Yeah, you just named it. You can go to high altitudes, i.e. in the mountains, and train. And this negative feedback system will work because you're at high altitude, and you can increase your hematocrit. And that's perfectly legal. So there's a thing called mountain training, and that is exactly right. And who said that? Who said that? Yeah, Christine. Joshua, training in Colorado. There is a reason why our Olympic training centers are in Colorado. Yeah. That's not an accident. Our Olympic training centers are in Colorado because of the altitude. Because of the altitude. It boosts their hematocrit. All right. Yeah. Christine went to the top of the documents. Yeah, and if we had more time, I'd explain all that stuff as well. But here's a quick anecdote. I don't want to drag this out. But I went to Colorado because I was considering teaching for the Air Force Academy there. And I took a trip with a couple buddies to Pikes Peak. Anybody been to Pikes Peak in Colorado? Gianni's going in August. All right, Joshua's gone there. Yeah, it's above 10,000 feet. So this is a true story. So I take this tram up the mountain, and on the tram, and I don't want to insult or offend anybody, but in the corner of the tram are two young men that I don't know what the proper term is now. I don't know if we're still using the term goth or emo. The ones that are, kind of dressed in black. Dressed in black, kind of pasty. So we get up to the top, and we get off the tram, and they go off to the side of the little store they had there, or the station there, and they light up cigarettes. And I'm with my buddy, and I just kind of elbow him. I'm like, watch this, watch this. So what do you think happened after they took 4 or 5 cigarettes, 4 or 5 drags from the cigarette? Passed out. Very good, Christina. They passed out. They're at high altitude, and now they're inhaling carbon monoxide. Yeah, but they both hit the ground. True story. And we went over there and helped them up and said, you don't smoke at 11,000 feet. Okay, so there's physiology for you, practical. All right, let's move on. We covered that. This is a big essay question, folks. This is worth 6 points on your test. Know it. You know the whole life cycle. You're going to tell me how the bone marrow differentiate. When you go back here in the bone marrow, the stem cell differentiates into those 4 phases. It enters the bloodstream. The average red blood cell, if it's lucky, lives for 120 days. Once it's worn out, remember, it doesn't have a nucleus anymore, so it can't repair itself. So if it gets damaged, it's done. So when it gets to the spleen or the liver, there are going to be big old mean macrophages that are going to engulf the red blood cell, break it down, well, again, efficiency, Tolani, efficiency. I don't need a lot of words. So again, I would start with, just to give you an example, all right, efficiency is everything. Yes, Jennifer?
Can we just write them like step by step, like what's happening?
Oh, yeah. Okay, listen to what I'm about to say.
Okay.
So I'm going to ask you about the life cycle of a cell. You're first going to say, boom, hypoxia, low O2, kidneys release EPO, stimulates bone marrow. Boom, done. Then you're going to go and say, we finally got rain here. Everybody got that part down. All right. Then you're going to say, EPO stimulates the bone marrow, and my stem cell becomes a proerythroblast, then an erythroblast, then a reticulocyte, and then an erythrocyte. Boom, done. And then you're going to say the erythrocyte lasts 120 days, and then it's phagocytized, broken down. I recycle the iron. I can use the iron. I can recycle the globin part because that's just a protein made of amino acids. And the heme is the only thing I can't recycle. So the heme, which is a pigment, is going to be metabolized into bilirubin. Boom, that's it. So that wasn't that many words. Did everybody get that? You probably all heard of bilirubin. That's metabolizing your heme from your hemoglobin, and that will be gotten rid of. All right, you'll excrete that in bile in the feces. Any questions? Keep it short. Keep it short. All right, let's move on. So again, this is your study slide, but again, you want to reduce it. In fact, I don't even care that you explain the changes in the differentiation flow. Just say stem cell, erythroblast, reticulocyte, boom, you're done. Okay. Simple. All right, let's move on. I'm going to just kind of blow off this right here because, again, you're going to get this in your immune lecture on Wednesday. So just hold that thought and we'll cover it later on. You will have to know these numbers, but again, we'll cover them in greater detail on Wednesday and explain why these numbers would change, what we call a differential white blood cell count. So again, save that for Wednesday. All right, and then we have to stop blood loss. We have three things that happen when we have some sort of damage, a cut. We have a vascular spasm, platelet plug formation, and blood clotting. So vascular spasm just means that we're going to cause vasoconstriction. We're going to have vasoconstriction. When a blood vessel is damaged, those endothelial cells are going to release endothelin and try to constrict the blood vessel to reduce blood flow to the damaged area. Then we have a platelet plug formation. This is basically trying to plug a hole in the vessel. This is not the same as clotting, all right? Make sure you see this down here. Clotting and platelet plug work together, but they're not the same thing, all right? So the simple version of platelet plug formation is you have damaged, and again, we're talking small blood vessels. This isn't going to work for your major arteries, all right? If you cut your femoral artery, none of this applies. Do I make myself clear? None of this applies if you sever your femoral artery. Okay? So this is for small vessels. So you have a cut, you've exposed the connective tissue, and now we have collagen fibers there, and for the platelets just immediately adhere to collagen. Once they adhere, they set off a cascade of reactions. They release thromboxane, A2, serotonin, ADP. These are going to fire up and activate other platelets. Serotonin, and there you see serotonin has so many different functions. And thromboxane A2 are going to cause vasoconstriction. ADP, and yes, that's ADP, adenosine diphosphate, so it has many roles. So that's one phosphate less than ATP, and that's going to cause stickiness. It's going to make the platelets stick together, what we call aggregation. And hopefully we plug up the blood vessel. Now, in conjunction with that, we have blood clotting. That's a different thing. But they do work together. So we have the platelets aggregating, forming a plug. We also have clotting, which is the aggregation of the red blood cells themselves. Now, this is a cascade reaction. It involves calcium. It's just an activator. And then we have a series of clotting factors that we use. And so we have two slides here that say the same thing. This is the more aesthetic slide that shows you the prettiness of it all. And then we have the one that Katie made. Katie made this, which cleaned everything up. And yes, this is going to be on your test. So we have two pathways. Extrinsic, just damage to the tissues. So yeah, this question sucks. All right, so damage to the tissues, and you need to know these, and this is cleaned up, folks. There's actually way more clotting factors involved, but these are the primary characters. But there are more as we learn, and that's why they're not really in sequence in terms of the Roman numerals, because as we learn more clotting factors, we're like, oh gosh, we already used 8, now we gotta, anyway, that's not important. So you're gonna tell me extrinsic, damage to the tissues, tissue factor 3, clotting factor 7, a dash of calcium, and then we go to this complex that forms factor 10. Intrinsic just means damage to the inside of the cell, not outside. So damage to the endothelium activates 12, platelet factor 3. So again, it's going to be working with your platelet plug. And 8 and 9 clotting factors to again form our factor 10. And then this is the common pathway. All right, so they both come to here where factor 10 is going to activate prothrombinase. What does ACE mean, somebody, really quick? Now, what does ACE mean? All right, enzyme. That was a good thunder crack. So prothrombinase is going to convert prothrombin. This is circulating in your bloodstream as we speak right now. This is going to convert prothrombin into thrombin. And thrombin, these are all enzymes, is going to convert fibrinogen, which is, again, circulating in your bloodstream as we speak, into fibrin. Fibrin is a water-insoluble protein that forms a net to capture the red blood cells. Here's a cool picture. So there you see right here. Once we've stopped the flow or the loss of blood, we need to break that clot down. So we have another enzyme that's going to break down the clot and dissolve it. And we have to do that because we don't want any blood clots hanging around inside the blood vessel. Okay? A blood clot in an unbroken blood vessel is called a thrombus. That's bad. If it happens to break off, we have an embolus. So if we have a free-floating clot in the bloodstream and it lodges in the brain, we call that a what? Somebody? Very good, Joshua. That's a stroke. We literally plugged up the blood vessel. If that embolus lodges in the heart, we call that a what? Well, I just said that, yeah, it's an embolus. So, yeah, Deanna, yeah, that's a heart attack. We blocked the coronary arteries, and so that's bad, all right? So that's why, aspirin is considered I hate to use the word blood thinner because it's not really thinning your blood, because that means your hematocrit. But yes, it prevents clotting. Any questions? All right, the last 4 or 5 slides I'm going to leave for the immune system. I need to explain what an antigen is and an antibody and all that stuff. So we will look at these last 4 or 5 slides later on. Okay, we'll cover all this later. And all this other stuff about the pathophys is just gee whiz. That's not testable. So all this stuff about sickle cell anemia, any of the anemias, none of this is in any way testable. Now I need to ask, what is the difference between an aneurysm from a stroke? An aneurysm is when the actual blood vessel ruptures, which is pretty lethal. So if you have one of your cerebral arteries rupture, and obviously you're bleeding in the brain, that's different than blocking. Okay? Beautiful. All right, so we're about ready to wrap this up, but I thought I'd give you a little treat here. Who wants to watch a couple quick videos of what I did for a living? All right. So I was an aerospace physiologist. And let me get rid of this thing. And one of the biggest problems, I worked in the fighter pilot community. All right. And I'm going to go back to this slide. Let me pull this up for just a second. because I think you'll really enjoy this. All right. Everybody remember this? Getting blood back to the heart? Well, we have a problem in the fighter pilot world with G-forces. Who knows what G-forces are? No one knows what a G-force is? Okay, Joshua just says yes. Okay, measure acceleration, all right. And so, no, I don't know about I really don't know what that even means. So the gravitational force. So while we're sitting here on the planet Earth surface, we are exposed to 1G, the force of gravity. In a fighter jet, we can create, and this is probably going to crush a lot of you. Who's seen the newest Top Gun movie? Okay. Really? You guys haven't seen it? All right. Well, a lot of it is just pure BS. Okay, just saying that. I don't want to break anybody's heart, but it's just BS. All right. So in a fighter aircraft, you can make turns that create more than one G or one gravity of Earth. And this is where it's BS in this movie, all our fighter jets are limited to 9 Gs. None of our aircraft can do more than 9 Gs. So in the Top Gun movie, they go to 10 Gs. I'm like, that's not a thing. That's not that. It's impossible, actually. But anyway, that's not my point. So I want you to understand the physiology behind this. So if I'm at 9 Gs, I have 9 times the force of gravity on my body. The blood now weighs 9 times what it normally does, and so it's going to travel down. And if I don't do something about that, my heart is going to starve, just like we talked about with a choir when you lock your knees, but this is even more extreme. And then I'm going to go unconscious. In the military, we call this G-induced loss of consciousness, because you have so many Gs. Again, your body weighs 9 times what it normally does. So think about this in another term. If you weighed 150 pounds and you were doing 9 Gs in a fighter aircraft, you would weigh 1,350 pounds. All right? And yes, it hurts a lot. It hurts a lot. So we have to come up with mechanisms to try to get blood back to your heart and back to your brain. So we have a G suit. And that's why I put this slide up here. And again, this is all practical. I think we've talked about this. So a G suit is just a bunch of bladders wrapped around your legs that when you pull G's, they squeeze, they inflate, and they squeeze, and they push the blood back up to your heart. You see this in hospitals, right? How many of you worked in the hospital where they have the little bladders that go back and forth trying to keep the blood flowing? In a fighter jet, you have a G suit, which, like when you have surgery, very good, Andy. So a G suit, when you're pulling high G's, G suit inflates with bladders and it squeezes your legs and forces the blood back up to your heart. But that's not going to be good enough. So then you have to do what we call the anti-G straining maneuver, which means, and this takes lots of practice, trust me, it takes a lot of practice, which means you have to contract every muscle that you can possibly incorporate, your legs, your arms, your stomach, your chest, And again, it's based on this venous return thing that I'm looking at here. You want to squeeze every muscle you have to force that blood up to your heart and brain. And it takes practice. If you don't do it properly, you go unconscious and then you crash and die. Okay, with that little, so that was one of my jobs in the Air Force as an aerospace physiologist. I was responsible for training astronauts and fighter pilots to deal with G's. Yeah, that's hilarious, Katie. You're spot on. So we have a human centrifuge. How many have ever seen a movie that had a human centrifuge in it? Most of them are kind of comical. All right. So we're going to watch a couple of videos of what I used to do in the Air Force of training pilots and astronauts to deal with G's and to perform the proper anti-G straining maneuver. Are you ready? I think this is going to freak you out a little bit. And I think you'll appreciate the next time you go to an air show and you watch either the Thunderbirds or the Blue Angels. Okay? So first I'm going to give you a video that is a production that was made by the military for the civilian world to understand what's going on. So what you're going to see is somebody in the centrifuge. This is a big arm. Yes, real life memories. Yes. And if you're good, I'll show you my experience. All right. So you're going to see an air crew. He's in a big centrifuge. It's spinning around, simulating what you would do in a fighter aircraft or spaceship. All right, so here we go. Hopefully this works. I don't know how it works with the Blackboard. Can everybody see that? All right, so this is your little lesson. Again, this is going to freak you out a little bit.
G-force, this is what happens. It's called G-lock, G-induced loss of consciousness.
Can you hear me?
This pilot volunteered to go through G-lock in a high-speed centrifuge on the ground. At high G, blood is forced downwards, away from the head, into the lower part of the body. The brain is starved of oxygen, and blackout occurs in less than 5 seconds. Even when G-force returns to normal, it takes up to a minute to regain control. If he was flying a jet, this pilot would certainly have crashed. So dealing with high G is an essential part of pilot training.
Okay. Okay. So that was a movie that was made for public consumption. Now I'm going to show you a couple private ones. These are some of my friends. Like I said before, okay. You have to learn to strain every single muscle in your body. And that is exhausting, folks. It's truly exhausting. I can't tell you how hard it is. You do adapt like humans do. And so let me go to the next one. And so if you aren't in good shape and you're trying to do this work, you'll just fatigue out. So here we have one of my buddies from way back in the day. Let me show this to you. Excuse me, I'm around here. Boom. All right. So everybody can see it, the video? Give me a confirmation. Okay, so what you're looking at real quick is if you look on the right side, or excuse me, on the left side, you'll see that he's at 5 Gs. So he's at 5 times the force of gravity. Look at his heart rate. That's that baroreceptor reflex that we just talked about today. Excuse me. And his job is to strain every muscle in his body to get the blood back to his heart and his brain. It's not a pretty sight. Excuse me, I got the hiccup. Pretty sight.
Stomach tight, breathe. 1, 2, 3. Squeeze the muscles, keep the lights bright. Breathe. 1, 2, 3. Looking good. Squeeze all the muscles, keep it tight. Breathe. 1, 2, 3. Make sure the butt and stomach's nice and tight. Breathe. Stay tight now, stay tight. Get them all back. Get them back. Get them back. Hello? Good. How you doing? Good. Just sit up straight for me there. You're sitting all crooked. Okay, what was your light loss when you released the brake? I remember. You know what happened? Yeah, green went. The green went? You know what happened?
Okay. So more physiology. So when you are under these, his heart rate did. And that's that baroreceptor reflex we talked about. All right. When the blood pressure drops in your carotid arteries, that sends a signal to speed up the heart because we're dying here. All right. And another physiology thing is when you're under high Gs and that blood is leaving your brain, the first thing that you notice is you start going blind because the oxygen going to your eyeballs, the retina, leaves first. Because again, remember this was an AMP1. You have that ocular pressure, and so the fluid in the eyes are pushing the blood out. So the first thing you go. So when you're in the centrifuge training, they have this array of lights right in front of you. And you can actually watch as your vision starts tunneling, as you start losing the oxygen to your retinas. And that's an indication of what you're doing. So you have to do this anti-G straining maneuver. You have to strain all your muscles and you have to breathe in quick, short breaths. You don't want to completely exhale because your lungs aren't strong enough to expand under high G. So you keep most of the air in your lungs and do quick, short breaths. And that's the anti-G straining maneuver. Okay. And there's 2 types of Gs. The slow onset, where you gradually go from 1 to 9. So your body actually has a chance to adapt with those baroreceptor reflexes. And then there's a high onset, which is actually what you do in a fighter jet, where you're going to go from 1 to 9 in a couple seconds. So when you go through this training, like I did, you have to go through both. So you guys want to see little Ricky when I was just a young whippersnapper. All right, here we go. So this is my first training way back when. And no laughing. Like I said, I'm a baby here. This is a long time ago. God, long time ago. Here we go. Okay. And again, I cannot emphasize this was a long time ago.
It is ready. Power ready, please.
Everybody see?
Use your brake and hold it. Okay, we'll put your head back. Relax. 3, 2, 1, pressure. Here we go. This is a lot like a carnival ride right here. It should start you going up.
So again, on your left, you see the G's. They're going to slowly go up. So I started at 1. And look at my heart rate. It's going to slowly go up.
Lose 100% of those green lights.
Uh-oh. I don't know what happened here. Let's try that again.
It is ready. Power ready, please. Okay, squeeze your brake and hold it. Okay, we'll put your head back. Relax. 3, 2, 1, pressure. Here we go. This is a lot like a carnival ride right here. It should start you going up and to the left. Okay. Let's just stay completely relaxed. Wait till you lose 100% of those green lights.
Okay, guys. I don't know why this is freaking out on me.
Three, 2, 1, pressure. Here we go. This is a lot like a carnival ride right here. It should start you going up and to the left. Okay.
All right, hold on.
Let's just take
I'm a little glitch. Let me try this again real quick. Give me 1 second. I don't know why it's doing that, but all right, this should work. Okay. I think this will work better.
It is ready. Power ready, please. Okay, squeeze your brake and hold it. Okay, we'll put your head back. Relax. 3, 2, 1, pressure. Here we go. This is a lot like a carnival ride right here. It should start you going up and to the left. Okay. Let's just stay completely relaxed. Wait till you lose 100% of those green lights.
Guys, I don't know why it's doing that. It doesn't make sense. I don't know why it's stopping like that. All right, so for some reason that's not working. So we're going to go to The rapid onset, hopefully this works. I don't know why that didn't work. Because that really was kind of like the better physiology thing here. I don't know why that didn't work so well. So we'll just move on to so that was slow. That took me up to 9Gs really fast, or really slow. This one is what you would actually do in a jet, and it's not pretty because I'm just learning how to deal with this stuff.
It is ready, medical. It is ready. Final ready, please. Okay, squeeze the brake. Hold it. Elite concentrate down. 3, 2, 1, pressure. Wait for me.
Hold on. No.
Oh, for heaven's sake, should I give it a try?
I don't know why it's stopping. I don't know what's going on here.
Let me
Do
give
you
it
have my videos?
Of course I do, yeah.
Well, give it a try.
Okay.
Start with a slow onset. I don't know what's going on with my computer. All right, everybody see that? I'm not hearing the video though, or the audio.
How do I share the audio as well? So camera and microphone. Phone? No, I don't want to do that.
Sorry guys, I don't know what's going on here. We can't hear you.
I don't know how to share my computer audio as well. Where's the actual settings for that? Do you know? Does anyone know? Right, audio, special notification, browser pop-up. I really don't know. How do you share your actual audio, Rick? Do you know? Speaker volume is at 100%.
All right, maybe we can just wait till Wednesday, Katie. I don't know what's going on.
I will stop sharing and just send it to you directly. How about that?
No luck, Katie?
No, not really, no. But I can send them to you directly, I think.
Katie?
Yeah, can you hear me? Testing, can you hear me? Rick, what happened to your audio? Oh, this is a cluster.
Yeah, I don't know what's going on. I don't know why mine just stops at after 10 or 15 seconds in my videos. I've been showing them forever. You can't get the video to go, Katie? Katie?
Yeah, I just sent you an email to your work email address.
And
Yep,
okay,
and my Dropbox.
all right.
Do you see it?
Yeah, I got it. All right.
Click on that. Click on the link. It'll take you to my Dropbox. There are your 3 videos.
Okay, let's try it again one more time. Sorry, folks.
Share your screen. We can't see anything. You have to share.
Hey, I'm multitasking here. Just give me a second.
I'm walking here. Hey.
All right, here we go. Let me try this again. Let's hope this works. All right, here we go. Let's hope this works. Can everybody see this? Shit. Jessica says yes. Okay. No, Joshua, I'm old retired guy.
They want to put your head back, relax. 3.
Can everybody see this? All right. Okay, thanks, Katie, for bailing us out here. All right, so let's rock and roll.
2, 1, pressure. Here we go. This is a lot like a carnival ride right here. It should feel like you're going up.
And like I said, this is a slow onset, so I'm going to go slowly from 1 to 9 Gs. Physiologically, watch my heart rate.
Wait till you lose 100% of those green lights. At that point, you can tell me now. Start pushing with your legs, tensing your arms and stomach. You no longer maintain the lights like that. Okay. Then you need to start the grasp point.
And watch me transform into
Stay completely relaxed.
Rocky Balboa.
Focus on the red. Looking real good. Stay totally relaxed. Looking good.
All right, hold on. Some of you can't. See this? Everybody, we've been online for a while. Can everybody refresh? Might want to refresh. Refresh, because it should be there. I see. I can't do both, huh? Dang. Crazy. Okay. All right. See, I can't. Oh. Can everybody see this? Verbalize if you can.
Yes.
Out. Thank you.
Push with those legs. Should get the lights right back. Just up your arms and stomach. Keep building up with that until you can't maintain it anymore.
You see, I'm at 7 Gs now. Now, my heart rate's 116.
1, 2, 3. Out this. Push the legs really tight. 1, 2. Hold it longer. 1, 2, 3. Out this. 1, 2, 3. Out there. Work at it now. Push them out there. Get me. Out this. 1. Keep straining. Keep straining. Keep straining. Put it all the way down. Okay, you can relax. Go ahead, relax and enjoy this part. This is the fun part right here.
Oh, man.
Like that rolling motion now.
Oh, man.
Your stop.
Yeah.
That was a great job. Excellent. I'm not going to say a thing. It was perfect. Any light loss?
Oh, excuse me?
Did you have any light loss on top?
Not really. Just not. Okay. Yeah. Well, that was. Yeah, get me. Yeah, get mean. Andrea, people have died from passing out and crashing into the ground. That's why you have this training. Okay, so now that was just to see what my G tolerance was, and that now you're going to see what happens in a jet. Again, remember, this is my first time, so it's pretty pathetic. Thanks, Gabriel. Well, this is pretty pathetic, to be honest with you. For my second one. All right, here we go. Can everybody see? Oh, wait, no. Oh, God, I gotta. See, when I go to the screen, I can't see you guys, so just give me a second. And entire screen. All right, here we go. This is rapid onset. Can everybody see?
Yes.
Thank you, because like I said, all right, here we go.
Anesthesia. It is ready, operator. It is ready, medical. It is ready. Final ready, please. Okay, squeeze the brake. Hold it. Really concentrate now. 3, 2, 1. Pressure. Wait for me. Okay, get on it. Hold up. Hold it in there. Hold it. On top. Hold out. 1, 2. Hold it longer. 1, 2, 3. Out there. 1, 2, 3. Out there. There you go. Push with the legs. Really tight. 1, 2, 3, 2, 1. Coming down. Keep straight. Okay, relax. Whoa. That was better. Focus on that red light. The higher up you go, the worse it is going down.
No kidding. That's worse than going up.
Down, bring yourself right out of the seat. This time push stick forward, squeeze it.
All right, so that was my very first time in a centrifuge. You go back a year later, after you've been exposed to a lot of G's, and you're way more comfortable. So this is my second time. I've been flying jets, and it's not anyway near as bad.
Trigger and release. System is hot. 3 legs, big breath. Get out back. Quick energy. Like nice and tight. There you go. 1, just a little bit longer, stretch it out. There you go, breathe. Look real good, just working that rhythm. Like nice and tight. Bear down the legs, unload, let go of the stick, keep spraying it, let go of the stick, let go of the stick, keep spraying in, breathe. And go ahead and relax. You must have been having a good time up there. I have to tell you 3 times.
Yeah. So the next time you go to an air show and you see the Thunderbirds or the Blue Angels, yes, Joshua, you have to do that and be in a dogfight. So, like I said, the new Top Gun movie is cool, but it's total BS. That's not possible. First of all, like I said, all our jets are capped at 9 Gs. You can't go to 10 like Tom Cruise did. It's impossible because the computer on the jet won't let you do that because humans can't do more than 9. So the movie's fun, but I'm kind of busting it here. And the second thing is one more video, and this is what you really would look like in the jets, unlike the new Top Gun movie. So this is what you would really, really look like if you're actually flying. So now I have my oxygen mask on. It's filling my lungs up, so I don't even have to inhale. It's going to inflate my lungs for me. I have a vest that's going to compress my lungs to push the air out for me, so I'm not working as hard. Can everybody see this video? Let's make sure, because like I said, hold on.
Yes.
With all my equipment on?
Yes.
Until you went to Blackboard.
All right, here we go. Okay.
How long can you keep that up for? Like, how long is that sustainable?
You have to do it. Good question, Andy. You to be a fighter pilot, you have to do it for 15 seconds because that's how long it takes you to make a turn. All right. So if you can't do that for 15 seconds, then you're going to fly an easier aircraft in there. So you have to do it for 15 seconds. You have to be able to stay conscious. That's a big thing. And yes, we did lose, I haven't been in the Air Force for a while, but we did lose pilots that would go unconscious and fly into the ground. All right. So the standard thing is pull 9 Gs for 15 seconds. Exactly, Joshua. They put you in a transport plane if you can't do that. But I just wanted you to see, the application of physiology, because that's everything that was going on there was trying to keep blood to my heart and get my heart to put it to my brain. And it is all about contracting every muscle in your body, which, like I said, it takes practice, like anything else, where you have to squeeze everything, like the guy said in the video, your butt, your legs, your arms, your chest, your stomach. And that's all about squeezing those skeletal muscles and forcing the blood back to your heart. And yes, like somebody eloquently put, and you have to do this while you're flying a jet. All right. So, and once, and I'll probably mention this when we talk about respiratory. At no time does your oxygen mask come off your face. Only in Hollywood, so that you can see how pretty Tom Cruise still is, and his other folks. That mask never comes off in the aircraft. When you put it on, it stays on until the mission's done. And we'll talk more about that when we talk about the respiratory system. Any other questions? We're good? Hello? Jennifer?
I do have a question. So I'm looking on Blackboard and I'm looking at Lab 9. Which one is the one that you want us to do? Because I see one that you have like a list that you want us to identify the white blood cells. And then there's like a hemocrit lab, I believe.
I want you to look at the hematology one, Lab 10. Lab 9 is just identifying the red white blood cells.
Okay. Just Lab 10?
Yeah, and we'll talk about that on Wednesday.
Okay.
Okay. Anything else? Okay, have a good night. See you Wednesday.